If you cut off the blood supply to your brain, your neurons don't just, slowly power down like a fading flashlight.
No, not at all.
Right. Within like 120 seconds, the cells actually begin to show literal structural damage.
Yeah, it's incredibly
The
fast.
intricate tree-like branching structures that connect them, they start to physically break apart. So today we are looking at what happens at minute 121 and, well, the days and weeks that follow.
It is a terrifyingly short window of time, honestly.
Yeah.
Because neurons, they're absolute gluttons for energy.
Yeah.
They constantly need oxygen and glucose just to maintain their internal electrical charge. And without that constant flow of energy, the cellular machinery basically just panics.
It just shuts down.
Well, the internal balance of ions and water collapses, and that kicks off this really rapid cascade of cell death. But the story of a stroke isn't just about that initial destruction.
Yeah.
It is ultimately about a furious, really dynamic reconstruction.
And that reconstruction is basically our mission for this deep dive. We're looking at a highly comprehensive research paper by Timothy H. Murphy and Dale Corbett.
It's a fantastic paper.
It really is. And we're going to explore the awe-inspiring, almost sci-fi ways your adult brain physically remodels itself, like down to the microscopic synaptic level, just to regain function after a major trauma.
Yeah, it's wild how it works.
But to really get into this, we have an expert with us today to help guide you through this complex landscape.
Yeah.
Because you really have a knack for connecting the microbiology to the macro behavior.
Oh, well, thank you. I try my best.
Okay, let's unpack this. Because to really understand the remodeling, we have to establish what ground zero actually looks like. So when a stroke happens, can you explain the difference between the core of a stroke and that surrounding area, the penumbra?
Sure, yeah. So the core is the central area where blood flow drops to near zero.
Okay.
And the cells there die rapidly. That damage is irreversible. But the real battleground is the tissue surrounding it, which we call the penumbra.
And that's like a halo around the core, right?
Exactly. It's this halo where blood flow is significantly reduced, but it's not completely cut off. So the cells in this area are injured. Like you mentioned, they might lose some of those tiny branching connections, the dendrites, but
they aren't dead yet.
right. If blood flow is restored, this region is totally salvageable. It's essentially stunned. And that surviving tissue in the penumbra is exactly where the biological seeds of recovery are actually planted.
Which I guess brings us to a harsh reality about that dead core tissue.
Yeah.
Because once a specific area of the brain is dead, the precise motor engram, which is like the actual physical memory trace for a specific physical action, that's permanently destroyed.
Completely gone. Yeah.
So when we see a stroke patient, eventually regaining the ability to reach out and grab a cup of coffee, it looks like recovery. But the research argues that true recovery is actually exceptionally rare.
Yes. Because true recovery, in a strict neurological sense, would mean that the exact pre-stroke motor patterns have magically reappeared.
Like the identical neurons firing in the exact same sequence.
Exactly. And that almost never happens once an anagram is lost. What we were really observing is behavioral compensation.
Behavioral compensation.
Okay. Right. The brain hasn't repaired the broken circuit. It's engineered a complex workaround to achieve the exact same goal.
And the paper illustrates this beautifully with an analysis of rats, right? Rats that have stroke-impaired forelimbs.
Yeah, this study is so cool. So researchers place a food pellet in front of these rats, and to the naked eye, the rat just reaches out and grabs it.
It just looks normal.
Right. It looks like they're just using their paw normally again.
Yeah.
But when the scientists ran a high-speed, like frame-by-frame kinematic analysis, they realized the mechanics of the movement were entirely different.
Wait, entirely different. How?
Well, the rats weren't using the fine motor control in their wrists and digits anymore because those specific neural circuits were gone.
Oh, wow.
Instead, they were executing massive postural adjustments, like the analysis showed them physically rotating their trunk or twisting their body angle to overcompensate. Exactly. Hiking up their shoulders to thrust the whole arm forward.
Yeah.
So they achieve the same goal, retrieving the food, but they are utilizing a completely different set of muscular strategies to get there.
It makes me think of like a sports team that loses their star Hall of Fame quarterback to a season-ending injury.
Oh, that's a good way to look at it.
Right, because you don't just magically clone a new version of that exact same player. That would be true recovery.
Right.
Instead, the coach looks at the remaining roster and just completely rewrites the playbook. They start relying heavily on the running backs, the tight ends. The team might still score touchdowns, But the underlying mechanics of the play are completely different.
That analogy holds up perfectly, actually, when you look at how the brain rebuilds its networks.
Yeah.
And it really clarifies a major public misconception about stem cell therapies.
Oh, yeah, stem
Yeah,
cells.
because people often assume that injecting stem cells means we're basically putting a new star quarterback on the field.
Right.
That the stem cells will just, turn into perfectly wired replacement neurons that drop right into the old circuit.
But the current scientific consensus from the text is that stem cells don't generally replace those lost neurons, right?
No, they don't. Instead, they act more like a chemical support system. They release these powerful growth chemicals called neurotrophic factors.
So they're basically flooding the brain with fertilizer.
Exactly. Fertilizer to promote the sprouting of new connections and like the growth of new blood vessels in the surviving tissue. They are just helping the existing surviving players execute that new playbook you mentioned.
Okay, but if the brain has to physically route new electrical signals to execute this new playbook, where is the traffic actually going? I mean, if the primary neurological highway is destroyed, how do the commands reach the muscle?
Well, this gets to the reality of how diffuse and redundant the brain's connectivity really is.
Yeah.
Because we tend to visualize the brain as a very simple point A to point B machine.
Like left hemisphere controls the right hand, full stop.
Right. But the brain is actually this spatially distributed computational network. It's full of redundancies.
Huh.
For example, while the left side of the brain primarily controls the right side of the body, there are actually these uncrossed hidden pathways that run from the left brain straight down to the left side of the body.
Ipsilateral pathways, right?
Yes, ipsilateral pathways. And
Yeah.
normally, they are just sitting there very quiet, not really doing much. But after a stroke, the brain can tap into these latent backups.
Okay, here's where it gets really interesting. Because the authors highlight this study done on raccoons to demonstrate these hidden networks. And it completely shifts how you think about brain mapping.
It really does.
Yeah.
Because raccoons, of course, have incredibly sensitive front paws.
Right, they're always washing things and fiddling with stuff.
Exactly. They have very distinct, highly developed representations in their sensory cortex. It's almost like human hands.
Okay.
So their somatosensory cortex, that's the part of the brain that processes touch. It's mapped out very clearly, like digit by digit. It's highly organized.
And so the researchers, they stimulated a specific digit, right? Like the raccoon's index finger.
Yes. And exactly as expected, the primary sensory area in the brain dedicated to that specific finger lit up with strong electrical activity.
But then when they looked closer at the surrounding areas, they found that about 20% of the neurons in that specific index finger area were also generating these quiet subthreshold potentials when entirely different body parts were stimulated.
Right, 20%. They were picking up this faint electrical hum from neighboring areas.
Wait, what exactly is a subthreshold potential?
Oh, it just means the neuron is receiving a signal, but it's not quite strong enough to trigger the neuron to fully fire and pass the message along.
Like a whisper.
Exactly, it's just a whisper.
Okay.
But the reason this matters to you, the listener, is because it proves your brain is filled with these latent circuits lying completely dormant. Your index finger neurons are secretly listening to whispers from your other fingers or even your wrist.
Wow. But under normal circumstances, the main signal coming from the index finger is just so loud that it drowns out those whispers.
Precisely.
But when a stroke destroys the main signal, suddenly there is silence. And the brain can start turning up the volume on those background whispers.
Yes. Over weeks and months, it physically strengthens those weak, diffuse connections to bypass the damaged area entirely. It recruits those whispering pathways and reinforces them until they become the new primary highway.
Okay, but strengthening a connection means physically growing new microscopic wiring.
Right.
Like axons have to sprout, dendrites have to branch out and form new synapses, and that requires physical space.
Which brings us right back to that surviving halo of tissue we mentioned earlier, the penumbra.
This battleground.
Exactly. Because the adult brain's sensory map is, well, it's like prime Manhattan real estate.
Oh, definitely.
Every single millimeter is occupied and highly valued.
Hmm.
If a building gets demolished, due to a stroke, the neighboring properties don't just sit there. They aggressively expand into the vacant lot. It is a total hostile takeover. And modern imaging techniques actually allow researchers to watch this happen in living mice.
Wait, really? How?
They use specialized voltage-sensitive dyes. These dyes literally emit light when a neuron fires an electrical signal.
That's amazing.
It is. It allows us to watch the brain's activity map shift and morph in real time.
And what they see is that when a stroke wipes out the primary sensory cortex for the forelimb, the surviving tissue right next door doesn't just stay in its lane.
Nope, not at all.
That neighboring tissue might normally handle signals from, like, the hind limb or the whiskers, but it actively starts relaying the forelimb's sensory signals down to the motor cortex. It physically rewires its own function to take over the lost territory.
Yes, and what's fascinating here is that in the animals that successfully recover their physical movement, these newly remapped signals actually look fundamentally different.
Different how?
They last noticeably longer in duration, and the electrical activity spreads much further across the surface of the cortex than normal, healthy signals do. It's as if these newly forged circuits had to shout a little louder and sustain that shout a little longer just to ensure the message successfully crosses these new, unconventional pathways.
But the brain can't just allow unchecked chaotic growth, right? I mean, if every surviving neuron just wildly sprouted connections in every direction to claim real estate, you'd have a neurological
It's
disaster.
complete chaos.
You try to move your toe and your eye would blink because the wires got crossed.
Exactly, which is why there is an intense biological tug-of-war happening chemically inside the penumbra.
Okay, walk me through that.
So in the early days after a stroke, the brain is flooded with growth-promoting factors, things like BDNF and GAP43. These are specific proteins that act as fertilizer, signaling the long cables of the neurons, the axons, to sprout and reach out to new targets.
But eventually the brain has to establish order.
Right. It needs to lock in the most useful circuits and prune the rest.
So it starts releasing growth-inhibiting factors, which I love this. They are proteins with literal names like NOGO.
No-go is a perfect example. Scientists aren't always subtle with naming.
Clearly.
But these inhibitory proteins, they act like molecular fences. When a sprouting axon bumps into a no-go protein, it triggers a chemical reaction that stops the growth cone right in its tracks.
It just halts it.
Yeah, it essentially tells the neuron, stop growing here, stabilize the connections you already have. It is an incredibly delicate balance. The brain has to allow enough wild growth to form a new functional circuit, but then throw up the fences to prevent reckless, chaotic wiring.
Okay, so if there is a window where the fertilizer is flowing before the no-go fences go up, then timing has to be absolutely critical here.
Oh, timing is everything.
So if I have a loved one who suffers a stroke, when is the optimal time to start rehabilitative therapy? Do you let them rest or?
Well, the science strongly points to acting early, during what neuroscientists call a critical period. The paper cites this foundational study looking at rats undergoing enriched rehabilitation.
And enriched in this context isn't just like putting a rat on a tiny treadmill for 10 minutes a day.
No. They put these rats in complex, multi-level environments. They had running wheels, extensive social interaction, and these intensive reach training exercises.
So they were forced to use their impaired limb repeatedly just to retrieve food.
Yes, a highly stimulating, physically demanding environment. And the results of this study were profound. The rats showed massive improvements in their forelimb reaching. They basically regained a high degree of independence.
Wow.
But the key finding is that this recovery only happened if the enriched rehabilitation started at 5 days or 14 days post-stroke.
Okay, but what if they delayed the start of the rehab to like 30 days post-stroke?
If they waited 30 days, there's almost zero functional improvement.
Zero.
Practically zero. And more importantly, when researchers examined the physical brains of the 30-day group, the deep layers of neurons in the cortex, the ones that actually send the final movement commands down to the spinal cord, they didn't show any new dendritic branching.
So the physical remodeling of the brain simply didn't occur.
Exactly. The window had closed, and the inhibitory fences were already firmly in place.
That is a staggering finding.
It really is. And if we connect this to the bigger picture, a stroke is essentially forcing the surviving brain tissue to temporarily reopen a critical period of neuroplasticity that usually only exists in early infancy.
Right, because when you're a baby, your brain is hyperreceptive to experience. It wires itself constantly based on whatever environment you were exposed to.
That is exactly what is happening here. The sheer trauma of the stroke forces the local brain tissue to revert to a highly plastic, childlike state just to survive.
But just like in childhood, that hyperplastic window doesn't stay open forever.
No, it doesn't. Over the course of weeks, the inhibitory factors slowly ramp up, the biological cement dries, and the brain permanently locks its new structural layout into place.
Okay, so during that golden window, when the biological cement is still wet, what are the actual rules the brain is using to decide which wires to connect? I mean, it's not magic. There has to be a physical mechanism driving this.
There is. The brain relies heavily on two fundamental learning rules to guide this rewiring.
Okay.
The first is called homeostatic plasticity. Now, homeostasis simply means balance. The brain constantly wants to maintain a specific baseline level of electrical activity.
Right.
But because the stroke destroys a massive amount of incoming signals, the surviving neurons in the penumbra suddenly find themselves sitting in absolute silence. So to compensate for this, they become hyperexcitable.
They literally turn up their own internal microphone.
Exactly. On a chemical level, they might actually embed more receptors onto their surface just to catch any stray neurotransmitters floating by. They increase their sensitivity because they are desperate for input. That's homeostatic plasticity, scaling up the volume to compensate for the sudden silence.
And that sets the stage for the second rule, which is Hebbian plasticity.
Yes,
The classic phrase here is neurons that fire together, wire together.
you got it. Once those hyperexcitable, ultra-sensitive neurons finally pick up a signal, and they fire an electrical pulse at the exact same time as a neighboring neuron, boom, they strengthen that specific synaptic connection between them.
So the homeostatic rule turns up the volume so they can hear each other, and the Hebbian rule physically wires them together once they start firing in sync.
It is a beautifully elegant two-step process, but it brings up a very difficult complication about how we approach physical therapy.
Right, and I actually want to push back on the timing we just talked about.
Sure.
Because we established that early intensive physical therapy was crucial for those rats to recover. But the sources also note that in some human trials, very early, highly intensive physical therapy can actually be detrimental to recovery.
Yes, that's true.
That sounds like a glaring contradiction. I mean, if the surviving neurons are desperate for input and turning up their microphones, shouldn't we force them to work immediately? Does forcing early activity somehow block that homeostatic volume dialing we just talked about?
This raises an important question, and that is precisely the risk. It highlights just how fragile this recovering ecosystem really is. The research suggests that forcing too much intense, repetitive activity on those circuits immediately after the trauma, say, within the first 24 to 48 hours, might actually interfere with the brain's natural ability to become hyperexcitable.
Meaning, we're essentially screaming into microphones that the brain is still trying to plug in.
That's a great way to put it, yes. If you flood the system with intense forced movement on day one, you might prevent the surviving neurons from naturally adjusting their sensitivity and finding their own new baseline balance.
The system gets overwhelmed.
Exactly. You have to respect the initial traumatic shock to the network. However, once that initial shock subsides, a few days in, you absolutely need the therapy to drive those Hebbian, fire-together, wire-together connections.
And that is exactly where techniques like constraint-induced movement therapy come into play. This is a fascinating approach where physical therapists will put a restrictive mitt on a patient's good hand, physically forcing them to use their stroke-impaired limb for daily tasks.
Yes, constraint-induced movement therapy is highly effective precisely because it prevents something called learned non-use.
Learned non-use.
Right. Without the mitt, the patient will naturally take the path of least resistance and just use their good hand for everything.
Makes sense.
But if they do that, the brain basically gives up on the stroke-affected limb. The no-go fences go up around a useless, silent circuit, and the chance for recovery is lost. By forcing the patient to use the impaired limb, you force coincident activity in the damaged circuits. You force those specific neurons to fire together so they wire together.
But again, you have to perfectly hit that golden window, like after the initial shock is worn off and the microphones are turned up, but before the molecular cement dries.
Incredibly tightrope to walk.
So what does this all mean? When you step back and look at all these mechanisms together, it completely shatters the idea that the adult brain is just a static, hardwired machine.
Oh, absolutely.
It's a dynamic, aggressively competitive ecosystem. When it takes a devastating hit, it doesn't just passively fail. It temporarily reverts to a childhood state of plasticity. It searches for hidden, redundant backup pathways that were just whispering in the background. And it engages in an all-out real estate war to literally remap the physical location of your senses and your movements.
It is the ultimate testament to biological resilience, really. But, there was one final, rather chilling thought pulled from the very end of this research that I think everyone listening needs to hear.
Okay.
Throughout this conversation, we've been talking about strokes as obvious catastrophic. You lose the ability to speak, or half your body is suddenly paralyzed. You know it happened.
Right, the siren goes off, the ambulance is called, you go to the hospital.
But there is a highly insidious phenomenon called a covert stroke.
Covert stroke.
Yeah. These are silent subcortical microstrokes. They don't cause paralysis. They don't cause a drooping face or slurred speech. There are absolutely no obvious physical symptoms when they happen.
Wow.
But they still destroy tiny pockets of neurons deep in the brain. And over time, the cumulative effect of these microstrokes gradually erodes cognitive function. And the reality is, covert strokes are several times more common in the general population than the noticeable strokes we usually think of.
So people are just walking around, going about their day, having these tiny microstrokes, and they have absolutely no idea.
Exactly. And the only reason they don't have obvious symptoms is because of everything we just talked about today.
Their brain is doing all the work.
Right. Their brain is utilizing its diffuse redundant connectivity. It is aggressively remapping real estate. It is silently relying on its own neuroplasticity to seamlessly route signals around these tiny microscopic roadblocks before the person even realizes a road was closed.
Your brain is constantly repairing the bridge while you are still driving over it.
Exactly. It's working tirelessly in the background to keep you functioning, masking the damage until it simply can't anymore.
That is incredible. And honestly, a little terrifying, mostly incredible. Thank you so much for guiding us through the actual mechanics of how this works.
It was my pleasure.
And to you listening, thank you for joining us on this deep dive. The next time you think of your body as a static machine, remember what is happening inside your own head. Remember the chaotic, beautiful scramble for survival at the cellular level and the hidden circuits keeping you moving. Keep questioning the incredible machinery inside your own head. We'll see you next time.